Hello, my name is Sean Bauersfeld and I'm with the Big Bang. Today we'll be going over proteomics for BENG 168. Proteomics is the comprehensive study of a proteome, defined as a collection of all proteins in a certain scope or system. We can investigate the proteome of a single neuron or a single red blood cell or even the entirety of the human heart. Proteomics also includes a study of subproteomes, which investigates the proteins present in a specific cell organelle, such as the endoplasmic reticulum. We study proteomes to investigate the structure, function, expression of proteins, and interaction between proteins. Proteomics is especially important for eukaryotic cells due to alternative splicing of genes. Proteins can be identified through use of a two-dimensional gel electrophoresis, where they will differentiate through two gradients. For the first, amino acids such as glutamate, histidine, arginine, and more contribute to net electrical charges on proteins. In the first gradient, the electric current will cause proteins to drift according to their net electric charge to their isoelectric point, the point at which they will be net neutral. For instance, arginine will drift towards the negative side. As the pH increases, arginine will lose its positive charge when its nitrogen is deprotonated into a neutral amine. In the vertical direction, proteins are separated by their molecular weight, enabling the separation of unique proteins of the same isoelectric point. First, sodium dodecyl sulfate, also known as SDS, is added to the proteins. The number of SDS molecules that bind to a protein is proportional to the number of amino acids in the protein. This results in a consistent negative charge to molecular mass ratio for all of the different proteins. The electric current applied across the second dimension causes the negatively charged proteins to migrate towards the positive end. Larger proteins will be slowed down due to frictional forces within the gel and will be closer to the negative end as a result. Once separated, proteins can be characterized and identified through mass spectrometry techniques. I will discuss two techniques. First, MALDI-MS. MALDI-MS begins with the addition of the enzyme trypsin, which will cleave the arginine and lysine amino acids in the peptide sequence of the protein on their C terminal. Once the protein fragments have been made, their presence and quantity is measured via use of mass spectroscopy. The results of mass spectroscopy can be compared with databases containing no quantities for different proteins and enable a researcher to find a match. ESI-MS-MS involves mass spectrometry analysis as well. Essentially, a protein will have one amino acid successfully cleaved on either the N side as a beta ion or purely on the C side as a gamma ion. The mass of each fragment will be analyzed, and the difference in mass that results from the cleavage of one amino acid can be used to determine the identity of that amino acid. Though, one caveat is that leucine and isoleucine have the exact same molecular weight and cannot be differentiated through this method as a result. The point of protein expression analysis is to determine changes in proteomes. Essentially how the quantity or presence of proteins changes for a certain disease. Now, an example could be a researcher who may be interested in comparing the proteome of a healthy cell or a cell with induced cancer. The before and after proteomes will be labeled and undergo 2D electrophoresis as described before. One way to differentiate the proteomes is dye. Cy3 and Cy5 are two dyes that can be added to the two proteomes separately and allow for comparison. Visual analysis of the 2D electrophoresis plate can show differences in protein presence and expression. Another method is Isotope-Coded Affinity Tag, which uses hydrogen and deuterium to differentiate between two proteome samples. The 8 hydrogen or 8 deuterium atoms will be attached to a linked chain that will bind to proteins via a reactive group. After the chains have been added to the proteins in their respective proteomes, in the avidin affinity chromatography step, all the proteins that successfully bind to the linker's reactive group will be captured and separated, since the biotin on the light or heavy chains of the linker group will enable differentiation from unbound proteins. Mass spectrometry then enables identification of proteins and protein abundance, where the 8 Dalton difference between the hydrogen linker and deuterium linker is very discernible. Lastly, microarrays. There are 3 types of protein microarrays: analytical, reverse phase, and functional. The first type are analytical arrays, which are used to detect or determine the quantity of proteins in a sample via adding a sample of multiple different proteins onto wells that hold many antibodies in place. Reverse phase are the opposite, where a sample of different proteins are arranged within wells and a single target protein will be added and analyzed to determine interaction. Lastly, functional arrays, which are used to determine how proteins interact with other proteins or molecules. That has been the presentation on proteomics. Our main source was the Molecular Biotechnology textbook, and thank you for watching.